physicists and theoretical computer scientists, and my talk today will not be a mathematical one. I'll just present some conceptual ideas and data. So recently it's become clear from evidence that mutation is affected by complex biological mechanisms that operate in the germline and by DNA sequence and structure. So what could this new knowledge, new empirical knowledge, does it have anything to tell us about how evolution works? So today I'll discuss three seemingly unrelated problems and how they may be connected together by revisiting the question of the nature of mutation. The first problem I want to talk about is the way we use chance in our explanations of evolution. In 1977, Jacob wrote, quote, "The probability that a functional protein would appear de novo by random association of amino acids is practically zero." But since 2006, people have found genes that look as though they arise from a junk, what presumably was a junk sequence, a non-functional junk sequence, and therefore all the mutations that are accumulated there must have accumulated, people think, without selection, until one day there were enough signals and coding sequences there that there was suddenly a functional new gene. How could this be, especially given Jacob's statement? The answer that's been in the air is that, well, maybe de novo genes are small enough, and given that today we know that there is much transcription going on in the germline, maybe by chance, once in a while, one of those minimal transcripts would acquire use by accident. But this is the Poldi de novo gene. It's 785 nucleotides long in its shortest spliced form. So could this really arise by chance? Let's talk about transposable elements.
Before you go to that, maybe you should tell what does the protein encoded by these sequences actually do?
Right. For the most part, we don't know. However, for Poldi,
particular one.
For Poldi, there's evidence, decent evidence that it is functional. If you disrupt it, it causes problems, and that's about as much as we know. We don't know more about the functionality of it, but it seems to be important, and that's enough for the point that it's an interesting gene.
Just to understand this, does the structural fold of this protein look like anything else in the world?
That's a great question.
Or does it even have a globular fold?
Those are wonderful questions, and I think we could have a full hour at least just discussing de novo genes, and that would not be nearly enough. Transposable elements, which actually are involved in the origination and evolution of de novo genes, were first described by McClintock in the 50s, and back then people mostly ignored it because there was no concept back then in people's minds that the genome it could be dynamic. But in time, the de novo gene started to be discussed, and in '76, Dawkins presented a way of trying to fit our understanding of transposable elements with the traditional way of understanding evolution, according to which transposable elements really are virus-like parasites of the genome, and so they're not really part of us, and in the sense, they don't disrupt our traditional understanding of the genome. And this view was elaborated by Doolittle and others in important papers that set the tone for understanding of junk DNA, which is riddled with transposable elements for many years. And they admitted that, occasionally bits and pieces of these genomic parasites can be co-opted for host use by accidental chance. But in recent years, it's becoming clear that the contribution of transposable elements to adaptive evolution is immense. For example, work from Günter Wagner's lab has shown that more than 1,500 genes have been tied together evolutionarily into a novel complex genetic network underlying a key adaptation, complex adaptation, that is the decidualization of the endometrium in placental mammals. And this happened by the movements of transposable elements and many mutations in them that interacted together in a holistic fashion. So a small piece of background that I may have not said. Transposable elements are these sequences that move around the genome or are copied and pasted around the genome, and maybe half of our genomes are composed of them.
In this example, the transposable element was carrying regulation around? Was that the...
Yeah. So it's the massiveness of this example and others, and many other cases, that tells us that these things are extraordinarily important for adaptive evolution. Could this be explained conceptually by the movements of genomic parasites and accidental mutations in them? Moving on to ENCODE, a large consortium of scientists that has been studied what was thought to be junk DNA intensively over several years, and announced in September 2012 that the majority of the human genome is active and needed, what they considered a reversal of 50 years of thinking, and that this finding will revolutionize our understanding of human biology, the human genome, and disease, and ultimately medicine, by greatly increasing the scope for genetical research that ultimately will provide us with opportunities for medical intervention. And shortly after, one of the main arguments that critics used in their criticism of ENCODE statements was that the amount of variation in junk DNA far exceeds the amount of variation that could be maintained by traditional natural selection. Nick Barton just talked about this very topic. And that therefore, all of this biochemical activity that ENCODE discovered, since it couldn't be subject to selection, it must not have a function. And therefore, most of it must just be chance associations of molecules.
Ali, what exactly do you mean by biochemical activity?
I'll say this very briefly. To my understanding, what this large group of scientists are saying is that they are seeing what looks to them like bits and pieces of biological machinery. Then they are assuming that they said that it has a function. And that's an open-ended statement. That's what it looks like to them. And then we come to, we bring theoretical arguments to bear on this topic.
They're saying that they see things sticking together. If they see something sticking to it, they say this themselves. That's what
So
they mean
in
by
biology, hold on one second. So in biology today, there has been a great debate over these results by ENCODE. People feel passionately about this. So you would find people that are very, they feel very strongly that ENCODE's findings are to a large degree irrelevant for our understanding of biology, and that they're misguided in a way, and that those are chance associations of molecules, okay? And then there are people who think that, well, maybe we don't understand yet everything about how things work, and perhaps there is merit to what these scientists are saying that they are seeing. So let me continue.
But can I just, there is an argument.
Wait, 1 second, let me finish my point. So if we look at very many observations of this sort, de novo genes, the contribution of transposable elements to adaptive evolution, these observations that I just mentioned by ENCODE, but also many other things, we have the following question. How much accidental chance are we invoking in our explanations of adaptive evolution? On the one hand, we say that in general, we have an explanation for adaptive evolution in the form of natural selection. By this, we imply that selection, not chance, must be doing the key part of the work of evolving an adaptation. But when we are faced with the evidence, and there are many pieces of it, very diverse, when we see any one piece, we then take liberty to assume that in addition to selection, there is also just as much extra accidental chance absent selection as we need in order to explain the event. And the case in point is the novel genes. So let me move on to the second problem. How do genetic interactions evolve? Fisher created a point of view where selection acts on additive variants, on alleles as separate actors. Each allele could be beneficial or detrimental in and of itself with little regard of the genetic context. That doesn't mean that Fisher was not interested in interactions. He was. But that was the perspective that emanates from his work. Sewell Wright, in contrast, foresaw the genes interact in complex biochemical networks, which today we know is obvious. And he thought that we must explain genetic interactions and complementarity and the evolution of complex genetic networks. So what did he propose then? How did he think that complex genetic networks evolve? He proposed his shifting balance theory, according to which in a small cell populations, alleles in multiple loci will not only appear by accident, but they, in a sense, will also spread by accident to fixation due to random genetic drift. Once they're all present, they can then interact fortuitously in such a manner that will allow them to serve as the basis for the evolution of a new adapted complex. So you can see the connection between the problem of genetic interactions and the problem of chance. Sewell Wright tried to solve this big problem of how genetic interactions evolve. And in so doing, he opened the door to our unrestrained, in a sense, unrestrained invocation of accidental chance in explaining adaptive evolution. So in a way, there have been two core ideas in 20th century theory for how adaptive evolution works. One is selection acting mainly on alleles as separate actors, and the other is the formation of the basis for new genetic interactions by pure chance. Besides this, there have been many models investigating the effects of selection acting on interactions within and between loci, but those are in a sense corrections or additions to the core ideas of selection acting on alleles and independent actors and the formation of genetic interactions by chance. In that sense, there has been no proposal for how selection acting on genetic interactions can be at the core of the adaptive evolutionary process, not in addition to something else that is basic. The third problem, what is the role of sex in evolution? Defined as the exchange of genes between individuals that lead to offspring that carry different genetic combinations from their parents. That's the evolutionary biological definition of sex, or one that you can use. According to this definition, sex or genetic exchanges really seems to be ubiquitous. It occurs in plants and animals through syngamy, and in fungi through the fusion of hyphae, and in bacteria by conjugation and other means, in fact. And importantly, it seems that obligate asexual species, they're not really species, but obligate asexuals are not only rare, they also appear to be headed toward extinction over evolutionary time. It seems as though they occasionally arise from sexual ancestors, and then ultimately they seem to go extinct. So asex is actually the derived condition. The ancestors are sexual. And already in 1957, Stebbins concluded from very extensive studies that obligate asexuals don't seem to be capable of evolving major new adaptations, but only of evolving minor changes on themes inherited from sexual ancestors. So sex requires so much effort, finding mates, applying the whole process, and it requires highly sophisticated adaptations today for its implementation, from human behavior to flowering and down to the molecular level and the machinery that is responsible for meiosis. So why sex? This has been a big question in biology. So one common answer that you might hear, not from experts in the field, but it's still a common intuitive answer that people say is, well, it's obvious. Sex generates a great number of different combinations of alleles across loci. It's what makes individuals unique. And since selection acts on individual variation, sex greatly facilitates evolution. The problem with this quick answer is that what exactly does it mean? Is it that sex will generate so many different combinations so that the best among them could be found by selection. But then the problem is that just as sex brings together different combinations, it also breaks them down. They're not heritable. And it is because of this that the sexual shuffling of the genes, it's because of it that we focus on selection acting on alleles as on single alleles, or alleles as separate actors. Either we focus on that at the core, or we can also look at pure chances driving the spread of alleles at multiple loci.
So why is that? Why are those the only two choices? Is it because we don't know how to deal with the interactions, or because it's some general argument?
I'll examine one possibility. So here's a challenge that I want to raise. The assumption that each allele has its own effect on the phenotype, in a sense, made sex not too disruptive to our theory, but also not essential. And this restricted us to asking whether and how it may contribute to subsidiary benefit on top of an evolutionary mechanism that works essentially without it. You can think of beneficial and detrimental mutations as arising with or without sex. You can think of the selection that we traditionally think of as happening in asexuals or sexuals, and ultimately being just fine. Perhaps not as good for a reason unknown, but ultimately fine. And one problem, all of these proposed subsidiary benefits require specific conditions that stand in contrast to the prevalence of sex in nature. So 3 problems. How much pure chance without selection are we invoking in our explanations of adaptive evolution? Another, how do genetic interactions evolve? And another, what is the role of sex in evolution? I mentioned in the beginning, now we know. Mutation is affected by complex biological mechanisms operating in the germline. Non-allelic homologous recombination, non-homologous end joining, transposition, complex enzymatic processes that affect point mutation. This is relatively recent knowledge. And this new knowledge raises yet another question. Why have we been believing that the mutation that drives adaptive evolution under selection is accidental? What is meant by accidental? Well, accident is a disruption. For example, one example of disruption is radiation. So here is an example of a fact. We know that we can blast Drosophila with X-rays and kill it by artificially induced mutation. That doesn't serve as evidence that the mutations that drive adaptive evolution under selection are due to radiation. What about copying errors? We hear that phrase so much. So much we hear that the mutations in the germline are copying errors. We hear it so much that it seems as though this is an obvious scientific fact that we all know. But copying error is not a fact, it's an interpretive phrase. The fact is that while some genes are being duplicated, others undergo genetic change. And to say that that genetic change, the cause of it, is copying error, is an interpretation. It's only one possible interpretation. In fact, it's based on a tacit assumption that the biological system is designed as though to make a perfect copy of the whole genome. In fact, we know that this tacit assumption is not right because the biological system doesn't appear to be designed to make a perfect copy, first of all, because it's sexual. So these are not facts, not evidence in support of accidental mutation. These are concepts, ideas that have accompanied 20th century evolutionary thinking. If it's not evidence that tells us that mutation is accidental, it seems that the problem is this conceptual issue, and we've had a good reason to think about accidental mutation. If we believe that the ultimate source of genetic variation can only be either accident or Lamarckism, then we're justified in choosing accident because Lamarckism cannot serve as a general level explanation for evolution. We don't believe that it would work in multicellulars. Even if some of us believe that it could work in specific cases, it doesn't serve as a general level explanation. If we think that these are the only two alternatives, we should choose accidental mutation as the source. But what if the mutation that drives adaptive evolution under selection is non-accidental in an essential manner for evolution, and yet is not of the Lamarckian kind? So that's the question that I ask myself already a few years ago, and today I want to tell you what I'm proposing. I propose that we revisit this question from square one. Okay? Think about the nature of mutation afresh. If the germline mutation that drives adaptive evolution is not accidental, then what would it mean? Then it's an outcome of biological processes that belong to the organism. And if so, then these processes are specified by the genes, just like any other processes in the organism, which means the genes interact not only in the determination of a classical trait, like the eye or the ear, but also in affecting genetic change. I wrote here determination of genetic change. I don't mean it to be deterministic. In fact, this goes back to Les Valiant's framework of evolvability, because really all I'm saying is that mutation is an output of a randomized algorithm. Now, if genes interact in the determination of mutation, then mutation is an outcome of a process that combines information from alleles at multiple loci and writes the result of the combination operation into one locus, the locus being changed by mutation. So this will be clear with the picture. Here are several different loci, and we see that cis elements and trans factors come together in a biological interaction. That part of the figure should be very familiar. It's basic to our understanding of molecular and cellular biology. What is not as familiar is this arrow of mutation here. Now, what does it mean? It means that mutation is a biological process that takes information from multiple loci and combines it and writes the result of this combination into a particular locus, a locus of mutation. And in so doing, it creates a piece of information that is heritable. It creates a new mutation, a new allele, which is this B star. And an allele is an elementary unit for the sexual shuffling of the genes. It is not itself broken down by this shuffling. And what this means is that even if this particular combination of genes, dark A, B, C, will never see each other again due to meiosis, due to sex. Even so, information from this combination lasts and is transmitted to future generations through the mutation that was derived from this combination. And this offers a new point of view.
Didn't you lose it by the mutations?
I'm sorry?
Didn't you lose the ancestral...
Yeah, the combination itself is no longer there,
Exactly.
but the mutation is there. And the key is to think of that mutation as an encapsulation of information flow. That's key.
Ali, since you got interrupted on this point, just continue for a second. I think it could be much clearer, your actual meaning could be made much clearer if you gave us an example of a molecular mechanism.
And wait 1 second. The less the interruptions, the more likely I will, and I would love to give you a link. So this does present a new view, which is that selection acts on the genetic combinations that are continuously generated by sex, and the combination of information from multiple loci by mutation transmits information to future generations from these genetic combinations. In fact, it transmits that information precisely in accord with the fitness of the individual as a complex whole. And this addresses the three problems from the beginning from a unifying perspective. The role of sex really is, after all, to generate a vast number of different combinations of genes. This role is fulfilled in conjunction with mutation being an outcome of a biological process, not simply accident. And this allows, for the first time, selection on individuals as complex wholes. No Lamarckism was invoked here whatsoever. So instead of the question of how much accidental chance is invoked in accounting for genetic changes, which doesn't seem to be amenable to the scientific method, we have a very different question that is amenable to the scientific method. What are the details of the molecular mechanisms underlying germline mutation? So let's notice something. again, I spoke of Les Valiant's framework, and once again, this provides basically a computational view of mutation and evolution, because mutation is an event that has multiple inputs from multiple alleles at different loci, and it has an output, which is the genetic change. Mutation is a computational event. Another thing, the output of one mutational operation at one generation, of course, can serve later as an input into the computation of another mutation. So for example, consider these two parents and an offspring. Here are the genes of one parent and the genes that it's going to transmit to its gamete. And here is a mutational event. These genes and the mutation are going to be transmitted to the offspring. Of course, there are multiple mutations in any one individual, we now know. And you can see that the output of a mutational event in one generation can be an input into the mutational event at another generation, which means that there is a network of information flow across the genome and over the generations, from any one gene into many genes, and from many genes into any one gene. And this information flow, I argue, is relevant for the evolution of genetic networks.
How can you store these mutations? They disappear. The mutations remain, but you cannot reverse them.
No. The point is definitely not to reverse anything. We're not trying to preserve the combinations. We're generating them again and again and again, but information is collected. In computer science, a picture of a network of information flow and computation would be a very normal thing. Let me try to get to the examples that Eugene asked for. Fitness determines the probability of information transmission from the genotype the individual carries. But now the individual is evaluated as a complex whole. This is an interaction-based framework. Okay? Interactions here are at the core of the process. There are no addition or correction to a process that's based on individual effects. And this involves many conceptual changes. Just very quickly, an allele has no value of its own. If we think about this carefully and deeply, it does change the way we understand phenotypic change. For example, how does phenotypic change come to be shared by individuals of the population? In the traditional theory, it's very easy to see. A mutation occurs in a particular gene in a particular individual. It provides a certain change in the phenotype, and then if that change is good, it spreads and brings that change along with it from the individual in which it occurred to everyone. Then another mutation could occur and so on. It's very easy to see how individuals would come to share macroscale phenotypic change. But under interaction-based evolution, well, alleles still spread in the population, albeit inconsistently due to the complex dynamics. Some, after a period of time, some will spread, some will go extinct, new alleles will come up. All those alleles that have spread after a period of time can provide a new amount of information that can underlie a trait, a phenotypic trait that is now shared by everyone, except that they did not bring independent pieces of the phenotype from the individuals in which they occur to everyone. They rather interact. And what this means is that the phenotype evolves on the level of the population as a whole. One would need to think about this for a while to see that this is a different concept and a different view. Again, what about the nature of the phenotypic change in terms of it being accidental or not? I spoke about mutation. Genetic change is being caused not by accident, but by computation. And what about the phenotypic change that it causes? Could that be accidental? Well, in the traditional theory, selection acts on independent alleles. An accidental mutation has its own phenotypic meaning that's complete at the moment of the appearance of the new mutation. All that remains for selection to do is to check whether this mutation is good or bad. And so selection is seen as an external judge of a phenotypic meaning formed at random before selection takes place. So traditional natural selection is thought of as a sieve. But under interaction-based evolution, the phenotypic meaning of a single allele that is spreading to fixation is going to depend on other concomitant alleles. But how the frequencies of those other alleles will change depends back on selection and interaction, which means that selection itself participates in the formation of the phenotypic meaning of an allele, which means that selection is not an external judge of a phenotypic meaning predetermined at random, but is actually an active participant in its formation, which means that the phenotypic meaning of a mutation is not random to selection because information from selection is already in it. So interestingly, under interaction-based evolution, the definitions of non-accidental mutations come together from above and from below, from the phenotypic level and from the genetic level. Both the genetic causes and the phenotypic meaning of the mutation are, in a sense, not accidental. And I would have liked to present to you a small sample of evidence, and I'll try to show as much as I can, and maybe we can take the 5 minutes of the discussion that was during my time. So rearrangement mutation is non-random. We know that it's caused by complex biological mechanisms, and they can cause insertions, deletions, translocations, inversions, duplications, and interestingly, this set of complex mechanisms still under much study. But all of these mechanisms we already know very clearly that they're guided to their places of action by homology between sequences and by DNA sequence and structural features. And also interestingly, if we think about the evidence that is unquestionable, we see that, different sequences that are involved in one mutational event are likely to be functionally related to each other, which is just an interesting deep fact about genetic change. That was rearrangement mutation, which is a huge category of things that you could read, a very large number of papers to try to get a sense of this new forest, a new world of evidence. But let's talk briefly about point mutation as well. The per locus mutation rate varies across the genome. And here is one example of an interesting point mutation. Some point mutations in humans occur in the cytosine of CpG dinucleotides. And putting together the evidence, it is beyond any question that the location of these mutations, their occurrence, is determined by complex enzymatic processes. In addition, we also know that there is adaptive relevance here because the locations are determined by methylation, and the change in the cytosine is going to affect future methylation, and methylation is important for the functioning of the organism through its effects on development. Unless you think that this is just some small, strange piece, these mutations account for 25% of all point mutations in humans.
So I don't understand what you mean by non-random. You mean that it's a non-uniform distribution?
Non-random would be non-uniform distribution. Okay. Non-accidental would mean that it's not an agent that's in a sense external. It's not radiation, it's not oxidative stress, it's not a copying error. There is a big system here that's evolved and continually evolving, and it is clearly determining or affecting locations of genetic changes and the nature of genetic change. See more. One second. See more examples, and you'll get a sense of how this works. Here's another quick little piece. Coincident single nucleotide polymorphisms. These are cases where in exactly the same nucleotide and related species like human and chimpanzee, you would see variation in the same location. The people who study this think that selection alone cannot explain this coincidence of variation in closely related species. So they have looked for sequence motifs in the location of these mutations to explain their occurrence. And this is not enough. They conclude that it seems that information from elsewhere must be determining this, which would fit with my theory, which predicts the determination of mutation is complex. Another example. We know today that meiotic recombination occurs in hotspots whose locations are determined by the combinatorial effect of the binding to transcription factor binding sites of multiple factors that interact with each other epistatically. And so we also know that meiotic recombination hotspots are also mutation hotspots. This means that in this case we have a somewhat detailed example of a mutation that we see is determined exactly in accord with the figure that I showed. Here's an interesting high-level fact. Today we know that there are multiple fundamental differences between the genetics of the germline and the soma. For example, transcriptional promiscuity, which people have tried to invoke statistically to explain de novo genes. Transcriptional promiscuity means that while the different cells of our bodies, each one might use a certain subset of genes that it needs, in the germline, products from across the genome are produced. Genes that would belong to any tissue could be transcribed in the germline. And from a traditional perspective, which of course did not predict or expect this, it's also, there is not a clear explanation. Why would this be, and how come there are adaptations that compensate for it and make it so that it's not destructive? But according to my theory, multiple genes must interact in the determination of mutation. This mutation must affect evolution. Therefore, it must occur in the germline. Therefore, we would expect that it involves genes of all categories of any kind of tissue should be able to interact in the germline to affect mutation. And so you would expect genes of all categories to be transcribed in the germline, which is exactly what we see, right? This is not the only fundamental difference between the genetics of germline and soma. There's also alternative splicing promiscuity. There's also global hypomethylation that's involved in transcriptional promiscuity, and a specialized RNA interference system that we know to be involved in complex regulation of transposable elements. And some argue uses them to implement genetic change. A lot of this is quite recent knowledge, and we spoke about the importance of transposable elements to evolution. So, everyone would agree with just a minute of thought that all these phenomena are of great importance for enabling evolution as we know it. The point is, from a traditional perspective, why is it that all of these phenomena take place precisely in the germline, precisely where their evolutionary potential is fulfilled? So for my theory, it makes sense. We have a way of addressing this because it predicts mutational writing activity in the germline. Quickly about the junk DNA and ENCODE debate. Again, one of the major criticisms was that only so much material could be subject to selection. And in a sense, repeating what Nick Barton presented in the previous talk, but saying it in slightly different words, when selection acts on genetic interactions, the change in allele frequency would be inconsistent in its direction. And so it may not, this is my own addition, it may not be easily distinguishable from drift. And so what looks to us through the lens of traditional theory is neutral genetic variation can actually be experiencing selection on interactions that may have much adaptive potential. And so work in progress with Eric Chastain, Kostas Daskalakis, and Christos Papadimitriou suggests that more genetic variation is maintained when selection acts on interactions as opposed to additive variance. And here is a nice piece. My theory predicts that there is what I call a mutation-writing phenotype. So just like the traits of our bodies are determined by complex genetic interactions that are evolved and continually evolving, so is mutation determined by complex genetic interactions that are evolved and continually evolving. And what follows is that just like the traits of our bodies should be more similar between more related entities, it would be more similar within a population than between populations, would be more similar within one species than between species, right? In the same way, mutational tendencies would be like that too. They would also be more similar the closer the relatedness. And for this, there is a great deal of evidence that is not really attended to or discussed or addressed by previous theory. So, for example, hemoglobin S mutation was the first mutation to be described on a molecular level. And it's become a quintessential example of evolution by accidental mutation natural selection taught in classes and written in textbooks. So many of us grew up thinking that by accident this mutation happened and the rest is selection. But evidence suggests that HBS arose at least 4 independent times in Africa. So one might say, well, okay, maybe the mutation rate is high enough. So it occurred many independent times and then was also selected independent times. The problem is that take the HBE mutation, which is a different mutation involved with malaria resistance. It seems to have appeared at least twice in Asia. But HBE seems to never have appeared, we presume, in Africa, and HBS never in Asia. So if the mutation rate is high enough, why is it that HBS appears multiple times independently in Africa but never in Asia, and HBE multiple times in Asia but not in Africa? So maybe it's just a statistical fluke or something else. The problem is that this is actually a general pattern. So when you look at the evidence, which you're not going to find in JTB or the journal Evolution or the journals we usually look at from an evolutionary perspective, you see this in journals like Blood, right? Because this is about malaria. And these people are collecting the actual evidence, and they have not been talking to evolutionary biologists, and this is what it looks like. So, for example, the deletion mutation, the alpha thalassemia deletion mutation minus alpha 4.2 appears to have appeared 6 times independently in Melanesia only, while a different mutation, minus alpha 3.73, has been found in other parts of Oceania only, and minus alpha 3.71 appeared to have arisen 3 times independently and is most common in the Mediterranean and Africa, and then the G2C mutation in the first intron of the beta-globin gene appears to have appeared multiple times in Asia and Melanesia, whereas another mutation appears to have arisen multiple times in people of African and Mediterranean origin, and multiple rare dominant beta thalassemia mutations in the 3rd exon of the beta-globin gene are observed specifically and recurrently in Northern Europeans, and then among them specifically the G2T mutation codon 121 of the 3rd exon appeared recurrently there, whereas other common mutations in that same place appear in other populations in the world. And so, okay, let's try to find a traditional explanation. Maybe the probabilities of origination of all of these mutations are the same for all humans, but in different geographical regions, there are different mixtures of the malaria parasite, causing different mutations to be fixed because maybe they provide different resistance tailored to different parasites, but we know that in neighboring villages in the malaria belt that have ethnicities that do not mix, we see very different spectra of mutations, even though the parasite are the same. So, okay, maybe the probabilities are all the same for all humans, but different costs and benefits due to different genetic backgrounds lead to the fixation of different mutations. Okay, this is nice, it makes sense, but it just doesn't give us a parsimonious explanation for the many, many facts of the situation. Take a look. We actually know that the genetic background and mutational mechanisms are affecting the origination of malaria-resistant mutations. Okay, so the alpha thalassemia mutations, for example, are caused by non-allelic homologous recombination that we know is a complex mechanism that depends on the genetic background of the organism. So we actually know that. Here is another example of a mutation that a beta thalassemia mutation that appears in a particular location where DNA structure exposes that location to it and makes genetic change more likely in it. So we see that DNA sequence and structure and complex mechanisms are affecting these mutations. And on the arguments go that basically show that an accidental mutation explanation does not give us a parsimonious explanation for all of these facts. Instead, the simple explanation is that mutation has a complex genetic component. And this has implications that are relevant to real-world important topics. For example, we talked about the differences between germline and soma. And we know that, or it was observed by a founder, one of the founders of cancer research, that cancer cells imitate germline cells, and especially sperm in many respects, including global hypomethylation, production of gonadotropin, expression of cancer testis antigens, downregulation of major histocompatibility complex and immune evasion, and power of proliferation of these cells, and other things. And this great cancer researcher, Lloyd Auld, concluded that cancer involves activation of a gametogenic program. So if Auld is correct, and if I'm correct, then the etiology of cancer involves activation of the evolutionary mutational machinery that normally operates in the germline. And interestingly, we know that cancer cells are exceptionally mutationally active. HeLa cells have been called non-human after 40 years of evolution in the dish. Here is the karyotype of the normal human cell and an example of the karyotype of a HeLa cell. They're very different. One last thing. I want to end with Darwin. In The Origin of Species, in the chapter dedicated to the laws of variation, in the first paragraph of this chapter, Darwin wrote that to say that variations are due to chance is a wholly incorrect expression, but it serves to acknowledge plainly our ignorance of the cause of each particular mutation. And in this chapter, he continues to provide many examples, many pieces of evidence that would not fit in any easy way with the idea of accidental mutation. For example, what has been under intense selection pressure in recent evolutionary times continues to vary intensely between individuals today. From the perspective of accidental mutation, variation is supposed to hit where it hits. Selection is supposed to act where it acts. And there should not be a relationship between the two. So why? And Darwin just stated from a long array of facts, he was convinced that this was a rule of high generality. And Darwin uses these words sparingly. High generality, he was convinced. From this observation alone, one could have inferred that something is guiding the variance because if things continue to vary today that have been under intense selection in recent evolutionary time, then it implies that there must also be parallel variation between related species, which we know is true from molecular evolutionary data and from Darwin's observations. But this parallel variation cannot continue forever because species should, in time, evolve apart more and more. So something must be there guiding the variance, and this thing must itself be evolving. Darwin called this generative variability, and I call it the mutation-writing phenotype. And so this is basically it. You can see much more details, many other kinds of evidence predictions in my paper, published in Biology Direct, 2013. And just keep in mind, the point is addressing many diverse facts that have not been addressed before from a unifying perspective. That's the strength of the proposal. And while addressing them from a unifying perspective, it brings up new directions for research, such as cancer research, new predictions, and so on. And that's about it.
I have a question.
Yeah.
Well, this sounds a bit fantastic in some sense, but genes want to get into the next generation. So genes now sort of influence how they get into the next generation. What is the genes can act selfish, right? If genes are the driver, what is the counterforce to account for fairness between all genes in a genome, saying we all go in this together?
Yeah.
You see what I'm saying?
So
it could go awry very quickly.
So unfortunately for right now, but fortunately,
Yeah.
in general.
Yeah.
You know, the question you're raising is really opening up, you know, a whole other huge
Yeah.
area, which is how does this, you know, what can we learn from this to this question of selfishness versus cooperation at whatever scale,
Yeah.
look at it, whether it's individual scale or the genetic level, and it has a lot to say about it, but time doesn't allow. But
Thanks.
thank you for the wonderful question.
Can I, so that was probably the best talk I've been to in quite a while.
Thank you.
But I think you're wrong to say it's your theory.
Go ahead.
Because, I mean, it's quite a new wheel, but I think you've reinvented it in a nice way. Because this is basically, you're talking about the evolution of evolvability, and there are many nice examples in addition to the ones you've given. So, the clustering of transcription factor binding sites in order to shape the genotype-phenotype map, or non-trivial neutrality, and all this kind of stuff. These are. So why do you call it your theory?
So right. So thank you for this wonderful question. I would have loved it if I was you know, taking a wheel and redoing it more nicely. That would have been a bit easier, and it would have been nice. Here is the answer. So evolvability, I'll tell you how, in what sense I agree with it, and in what sense I'm presenting something different. Okay, so the sense in which I agree with it is that evolvability, and this is not evolvability in your sense, you know. Evolvability exists. From my perspective, it is a fundamental thing. So I agree, it exists. The question is our understanding of it and how it comes up. So the previous proposals, you know, implicitly or explicitly, what did they say about the origin of this evolvability? I don't know. I would presume that at least implicitly, people think that you know, ultimately you have a source of variation that is accidental mutation under selection, and then maybe you bring to bear on this group selection at whatever level, or you do other things and you examine it with modifier theory, and you know, a lot of wonderful work has been done. But it's very different in nature from work that says that mutation is fundamentally a computational process of information transmission. Have you heard that being said in the evolvability ideas that have been proposed before, besides the one of Les Valiant's, which uses evolvability in a different sense?
I don't think that's true. So, for example, the work of Mark Toussaint,
Okay.
which talks about non-trivial neutrality. There, you have a situation where you're actually learning exploration distributions and genotypes that produce grandchildren with higher fitness to be selected for. So that's what the Altenberg was talking about. So this is various adaptations for structure.
So evolvability. You know, I read the evolvability, you know, literature. I don't recall the particular interesting paper that you're mentioning. I would love to read it too. You know, it is clear that it's a different literature than what I'm proposing. They do agree that there could be mechanisms influencing mutation. But the philosophical framework is very different. Here it's one that's based on genetic interactions and mutation as a computational information transmission event. This puts together empirical phenomena that have not been put together before in this way. The role of sex in evolution, genetic interactions, the way we use accidental chance in evolution. From this follows, you know, a proposed understanding of many different empirical phenomena and new predictions and new directions for research that we're raising. So that's really all. And
I think you've definitely got new empirical facts.
Yes, please.
So two things, but very concrete. One, you claimed in one of the slides that the notion of the role of chance in evolution is not amenable to scientific study, if I am not mistaken. Honestly, I think this is not correct. Well, it's not only amenable to scientific study, but it has been studied in a very specific manner, and in particular, as highlighted by Nick in his talk, in the theory of genetic drift, and importantly, the drift load, and equally importantly, what has been not mentioned in Nick's talk, that notion of the threshold recently introduced by Mike Lynch. So I think it's not only amenable, but has been investigated and specific bounds have been put in. That's one thing. Okay, and maybe I will be
Maybe I'll present and then the second. Yeah, so thank you for this wonderful question, because it allows me to make an important correction. What you said, in my opinion, is exactly right. So let me try to clarify and qualify what I was trying to say. Random genetic drift has been studied so much. We are trying all the time also to distinguish between mutations under selection and neutrality, but with the tools that we have had so far. So yes, in that sense, we are trying to study the role of chance. Okay. So what I meant is the following: when we observe phenomena that a side observer who is not encumbered with ideas that we carry with us since the 1930s, examples that such a side observer looks at and says, this is really strange, such as de novo genes, what do we do? Really, the response has been, okay, so that must be due to chance after all. And what I mean by this is that we're not putting a line, we're not putting a boundary that says, once we start invoking chance for these, please, take us aside and, don't pay attention. We're not putting that line. So when Jacob says in 77, there shall be no de novo genes, now we see things that look as though that's what they are. So okay, that's chance too. So there is no quantification, there is no measure. In a sense, this undermines our theoretical efforts, because we have all this extensive, population genetic theory that is so wonderful and so well developed, and yet it doesn't address that question that lies, behind this, at the basis of things, which is, how likely is this explanation? How likely is it for the de novo gene to appear by chance? We don't have a calculation, we don't have evidence, and so we presume that from what we see, that must be because. And that's what I mean to say that we cannot place such a line. No matter how many models we have, there is something here that is dangerous, because there is something that's irrefutable. We see the data that is strange, the de novo genes, transposable elements putting together networks, and we say, okay, there's some selection in there, and the rest is random genetic drift, and we explain. But we don't know if that's true.
May I continue? Another quick one.
Okay, so that's three more questions altogether. Yours plus three more. Okay, so finish it.
So this will be a bit of demagogy, but nevertheless. Another question. So you used, harking back to the previous question, you used the phrase "my theory" quite a number of times. Usual. Correct me if I'm wrong. Usual, my perception of theory is that this is some kind of equation, some kind of or set of equations, or system of equations, some kind of relationship that either fits the data or some kind of data or not. And if it doesn't, it's falsified and something else needs to be developed. So could you present some very hypothetical experiments or models that could refute your theory?
Yes, so first as to the definition of theory that you used, which is, present a certain equation, a certain model, and show, a quantitative prediction. And by that, you just defined theory. But that definition of theory, would mean that Darwin's theory of natural selection was not a theory. Okay. So okay, that implies that theories are not the only important things in science. So that implies that from your terminology, So from your terminology, you would have liked me to change the word theory into another word. My ultimate interest is to try to help the progress of science in the best way that I can. That's it.
So if you use their concept, I wouldn't ask a question.
So we'll defer this to this afternoon. Okay, so Lee.
So sort of in my framework, you have differences in the kind of composition of offspring you produce. You can think of those, that genetic variation in the different composition is within the realm of modifier theory. But so it sounds to me what you're proposing is basically smart modifiers that are varying something far more sophisticated than simple mutation rate or combination rate. It's a much more intricate variation in the production of offspring. Would you agree with that kind of framework?
You know, I mean, yes and no. So in one sense, you can think about it like that, as a very complex situation with modifiers, you know, affecting things by epistatic interactions, and not single modifier that has a single effect of its own. That's one important, you know, difference. And you know, there are other important differences, like the basic idea and the basic understanding that is presented, right? So, you know, we are talking here about information flow across loci over evolutionary time. So, you know, we need to capture that. I believe that Les Valiant's framework is extraordinarily interesting for us in theoretical biology because it offers new tools with which to try to capture such things. So, you know, in one sense, you can think about things in this way, but it won't be enough to capture what I'm proposing.
So as far as the applicability goes, I can't figure out where my theory, and I'm going to say my theory, both in Eugene's sense of theory and the regular sense of mine, in terms of how de novo genes occur, am I categorized as accidental? I mean, there's a huge
So I will let you answer your
flow.
question, but I'll remind you how you answered it yesterday. You said at some point that, you know, we now know that even whole genes can arise by accident. So maybe that's your answer to it.
Well, no, that was my I mean, under your philosophical definition of chance or accident, and forget about how I define those terms.
Okay.
In the way that you define those terms, where does my theory fit in?
Okay, so, I mean, if you want to hear that, first of all, you know, I'm fascinated by the work that you're doing. We obviously share a great interest in this topic, and I think that what you're doing is very important. You're producing all this, you know, evidence that I find extraordinarily relevant and important. I think that we differ in our perspective on the meaning and causation of this, where I would predict or propose at the general level that it's not simply a read-through error that allows for traditional selection to come in a little bit before the time when this product is experiencing selection as we thought it should, but that this data reflects more complicated events that take place over evolutionary time that in a general sense represent the flow of information across loci. If you want me to give you the details of this, I have no idea. The point is for science to go on and study this. I'll give you one answer. Let me give you a piece of an answer, okay? We know that the novel genes, we know that transposable elements are important for the novel genes. So that's shown by the data. They are more important for the novel genes than the papers currently even report on, because when you look at those genes more than before, you see yet more transposable elements underlying that activity. A transposable element, in one general sense, it is a flow of information across loci, right? So the question is, are we gonna address all of these observations as specific, unique questions where we say, let's think about the novel genes on their own and say, okay, maybe it's a read-through error that increases the probability for a new whole gene to rise by chance. And we will address the question of transposable elements on its own and say that, maybe we should conceive them as, parasites that move around and by chance they get adopted here and there. Or are these really pieces of much more complex and sophisticated machinery that is operating in the germline, that is affecting evolution and is part of evolvability in a way that we haven't thought about it before, but in a way that means information flow across loci that connects different empirical observations and opens up directions and predictions.
Okay, so two interesting questions.
Cancer connection you mentioned, I guess just wanted to make a point that for cancer initiation, I guess your comments are correct, but when the phenotype progresses into a higher invasive phenotype, we don't know the environments that they can, epigenetic factors are more important, the expression of the genes rather than actual genetic changes become important in cancer. But those are really highly correlated to environment and non-random. But so in cancer, we have to more clear about when. I mean, it's correct in cancer in general. It's correct. A lot of the effects are environment effect and non-random, and might be like half and half mutations. Like the change of phenotypes might not be exactly a mutation.
Yeah. So none of this, none of this, none of this interferes in any way with what I try to state, which is.
I agree with what you said. And one more comment. In the biological assessment of mutation, whether can be a chance or not. I'm aware of this in the context of like single cellular, E. coli. Drug resistance, so maybe like 2004, Robert Austin in Princeton, they can actually pick on the individual resistant cells and do the gene expressions and see when the mutation happens. And the mutations, most mutations are not very random, they're very correlated, always happen. Majority of mutations happen on the where it's supposed to happen and the drug resistant genes will change. So in supporting your idea, supporting it, just say.
So can we have the last question?
So I have a very concrete question. I don't understand most of the words being used, but I also don't understand the examples. Okay, so this is a concrete example of Lee's question, which is a yes/no question. We have an enormous number of DNA polymerases, 20 of them. They have very different profiles of errors. They're controlled by a large number of genes, and presumably the error rates and profiles of these vary from species to species. Is that an example of what you're talking about?
So once you phrase it in terms of error, you're introducing your interpretation into it.
I'm not introducing an interpretation. Is that an example or not? No interpretation, it's a yes/no question.
If you want to change the word error to change, genetic change.
Yeah.
These are causing genetic changes. These are causing, what I'm saying is that genetic change, and what I'm saying is that the origination of genetic change may not be accidental, and that might change fundamental concepts in our understanding.
It's a question, here's a specific biological mechanism, specific thing that goes on. It would seem to me to be in your framework. It's a bunch of genes that come together. They determine which polymerase is used.
Yeah,
those polymerases have changed over time, and they can make another.
Well, anyway, I think thanks very much for your talk.